Thank you for being here. My name is Anca Rath. I'm a consultant in infection control at the University Hospital of Regensburg in Germany, where we have implemented molecular surveillance for all multi-drug resistant pathogens we diagnosed at our hospital starting in 2019, 2020. And today I will present to you the results we've had on Pseudomonas aeruginosa, strains defined as multi-drug resistance level 4 by German definition, which means resistant to piperacillin, ceftazidime, cefepime, meropenem, imipenem, and ciprofloxacin. I'm presenting to you the period of 2019 to 2023, where we had 132 patients diagnosed with this pathogen, of which we had 99 patients with isolates in our collection. We sequenced those on an Illumina platform, and these are our results. Well, some results you might have expected, like a great diversity among the strains, which is depicted in figure 1. You see the long lines, which indicate a high or a large distance from other strains. But among them, we have some clusters and some clones which are more frequent. We have the so-called high-risk clones, sequence type 111 and sequence type 235. 111 is here, so we have a few isolates of those, and 235 down here in green. But while they are closely related, they are not close enough, so we would have suspected from an infection control point of view an issue at our hospital. A less known clone is sequence type 309, which is depicted up here, and where you see it's about 20 isolates we've had of those, which was very concerning to us, and we decided to go next level, because surveillance is not about collecting data, surveillance is about using the data for daily purposes. So the first thing we did was to look when were these isolates found at our hospital, which is depicted here in the timeline. We took the different clusters and divided them in a different timeline. We have the big cluster up here in yellow is depicted on the top row, as you can see. Right here we have a small collection of isolates. They could be traced back to one ICU, most of them at least. But as you can see, they are focused in 2021. And we had looked at the data in 2023 with more time to take and look at it. So tracing back was not quite possible back then. And we left it at that as we didn't have any more cases. In the second row, where you see a less spectacular result, we have singular cases every half a year, every year, and a total of 6 cases. But the last one was found by the end of 2023. That's why we decided to go for it and dive into the data. By the beginning of 2024, we initiated screening on a department where we could trace back some of the pathogens. Most patients had been transferred away from the department when they were diagnosed with the pathogen, but that was the only connection we could find. We screened the patients and we did environmental investigations, and we found a few patients with multi-drug resistance, Pseudomonas of the same resistance pattern phenotypically, but genotypically it was a very different strain, and we also found it in one sink across 2 departments with over 30 sinks that were sampled. So that was why we had found these pathogens and could intervene. We had not found the source for our other pathogens. We maintained screening for a few months. We didn't find any more cases, so we ceased to do that. Later on, cases started reappearing about summer at this time. So we started doing screening again, we reinitiated environmental investigations, and finally we detected that the sinks in the nurses' bay also had the same genotype. We did several courses of disinfection. We could not eliminate the pathogen despite that, so we were quite sure that was our source. We prohibited the use of these sinks, and when you can look down here at the genotypes and at the pairwise comparison depicted as a minimal spanning tree, you see that the blue drops indicate where the sinks are. We also had some sinks in the patient's rooms where one course of disinfection was sufficient to eliminate the pathogen, but they kept reappearing except for the nursery spray where you could not eliminate the pathogen. We prohibited the use of those things and went on to remove them, including a few meters of the pipe for different reasons, also because they had to be renewed. And ever since, we have not seen any cases of transmission of this genotype. So it's almost 1 year later since we prohibited the use of the things, and it seems to be very efficient. What we are currently doing is, even though we have great genetic diversity among the strains with over 30 different sequence types and over 70 types of complex types, we are looking into plasmids. We started doing long-read sequencing as well to see whether there's more connection between the strains there, and this maybe could explain the great number in detections with maybe plasmid transmission. We are still working on that. First results indicate that when plasmids are there, they are related to biofilm formation, oxide reductase, so adaptation to the environment, but the results are still to come. Stay tuned. So thank you for listening. I would also like to thank our team in infection control who did all the investigations. We would like to thank the team in the department for all their efforts, which were prolonged for over a year doing screening and maintaining a higher standard in infection control to prevent transmission of the pathogen from the respective sources, and all the team in the lab who did the sequencing and enabled us to do such data analysis. And thank you for listening and your interest in our topic. Have a nice day.